Okay, hello there. My name is Grace McCormack, and this is part one of a three-part lecture on the origins and diversity of honeybees in Ireland. And this first part is just some of the background to that lecture. And the reason for three lectures is just the smaller file size for making available and for sharing the information. The reason for this lecture was because it became evident to me when I was trying to remove hybrids from my colonies where I am in Galway, that some of my new virgin queens came back with mated with non-native drones. So I was getting a mixture of hybrid offspring in my colonies, of my new colonies. And I asked in the local association about what kind of bees people had in the area, and did they know any area around where it was easy to breed native bees, i.e. Apis mellifera mellifera. And by return, quite a number of beekeepers replied that they didn't know what they had. So, and I shared some pictures of my hybridized bees, and some thought that they had some like that, and then they had also had some black colonies. So this information really, I suppose, isn't widely available to beekeepers. So I thought it might be useful to prepare a lecture, and this lecture, all three together, was given as a webinar for the local association, but it was opened up more widely, and then to the Irish Beekeeping Associations as well. And so I decided to re-record it in three parts and to make it available. But the rationale was really so that people understood what kind of bees we have in Ireland, what kind of honeybees we have in Ireland specifically, just to inform and help us, I guess, manage our native bees in the face of the ability of people to bring in non-native bees, and particularly for beginners who may not know the difference, and other beekeepers also who are looking for bees and who don't know that there's a difference between native and non-native. And so just to maybe consider some of these elements when getting bees. So, oops, I'm going to put my oh, sorry. There's a pointer. Okay, next slide. Okay, so I am an evolutionary biologist. I use, I suppose, fundamentals in evolutionary biology to look at relationships between organisms through time. And this is also called the area of systematics. So looking at biodiversity and the diversity of organisms and the relationships between them. And I suppose fundamental to this is the idea of Darwinian evolution. And so everybody really knows who Darwin is, and his theory of natural selection and origin of the species. And so his idea that basically actually all life on Earth descended from one ancestor back in the day. And his theory of how this happened and how diversity can be explained and different species can be explained is through this idea of natural selection or survival of the fittest. And back at the time of Darwin, he wasn't familiar with the area of genetics. While he could see that change was happening over time, he didn't know how that was happening. So what were the traits that were he didn't know about inheritance and about DNA. And subsequent to that, we have, of course, huge expansion in our understanding of genetics now. And when you marry genetics with Darwinian evolution, you get what's called the modern synthesis. And so we can study evolution using DNA because, anyway, so evolution itself can be measured as changes in inherited traits over time across generations. So I think of this as, we can track evolution through time by comparing and analyzing particular characters or traits that organisms have. And these traits that are shared in different ways across organisms can inform us about how closely related these organisms are and maybe where or what they evolved from. It's important to note that the traits that we use to do this are traits that are inherited. And so that's just depicted here in the figures, whereby we have these species of honeybee that are all species of bees that are all related and all belong to the genus Apis. So the Apis is a genus, so it's a group of related species. And so these various species have arisen from that ancestor, and we're interested in Apis mellifera, that western honeybee on the top here. Some of these pictures actually were taken by John Breen of UL fame. So some fabulous pictures here of A. florea, A. dorsata, and A. serranae from his travels. So the idea is that Apis mellifera, so the honeybee, Western honeybee, evolved from an ancestral species of honeybee, as did these other bees. And so Apis mellifera is related to these bees, but can't reproduce with them. So I said that the traits that you can measure evolution with are traits that are inherited, and traits are inherited by DNA. And DNA is a molecule that's present in all of the cells of all organisms that really contain the blueprint or all of the instructions for the development and functioning of that individual. And so it's just a chemical that has particular structure and that resides in the nucleus of cells. And so the figure here in the top left shows you a cell, and inside that cell you have the nucleus, and inside the nucleus we have something called these chromosomes, which are structural entities comprising of DNA. So they're comprised of DNA that's tightly packed, tightly coiled in together, and protected by proteins to keep them there and to protect them because of the important function of DNA. When you unravel out these chromosomes, you see the double helical structure of DNA, that type of twisted ladder that is known as the structure of DNA. And if you open that out further, you see the structure even closer of that double helix. And you see that the double helix is comprised of 4 chemical types. We call them bases: adenine, A; thymine, T; guanine, G; and cytosine, C. And these represent the DNA sequence that you often hear people talking about, A, T, C, and G, that make up the DNA. And these are held together by a sugar phosphate backbone, so that the sugar phosphate make the edges of the ladder and the bases form the rungs. And you will see that typically in DNA, adenine is always bound to thymine and guanine is always bound to cytosine. And this is because of the chemical structure, and there's their natural pairings. And there is a bond that connects the adenine and thymine, and therefore and so on down the ladder, and connects those 2 halves of the ladder. But if you set DNA at a high temperature, for example, the bonds between those break and the DNA kind of comes apart into 2 halves of the ladder. So when cells replicate to, make up, when your hair cells and skin cells replicate, but also when gametes are formed, these chromosomes and the DNA divides. So it also replicates, basically. And so DNA gets replicated in the cells on a regular basis. And to do that, those bonds, the DNA separates. So the bond between the bases separates to give you the 2 halves of the ladder. And then each half of the ladder is used in order to copy, to make the other half. So that once the replication has happened, the ladder divides into 2, and then each half is replicated so that you get 2 ladders again. And so when that happens, sometimes mistakes get made. So sometimes instead of placing a thymine, it'll place an adenine, or it'll place a cytosine, and this mutation happens. And most of the time it gets corrected. We have enzymes that can correct these mistakes, but sometimes it doesn't get corrected, and you get mistakes or differences happening between, say, parent and offspring. So in the generation of sperm or the generation of an egg, in the formation of the DNA that goes into those gametes, you can get mistakes made, and therefore the egg will have a slightly different DNA sequence from the mother. And so, as I said, in most organisms, we have repair mechanisms in order to fix that, but they're not perfect. So over time, you do get mistakes happening in DNA replication that then we can sometimes see as visible in the next generation. So we have 2 terms here that are important. One is called the genotype and the other is called the phenotype. And firstly, the genotype can be defined really as all of the DNA present in an organism. So it's the type of DNA present, or the genetic makeup of an individual. So you can see from the top picture there again, the cell, the chromosomes, and then you unravel out the chromosomes, you see the double helical structure of DNA. And in that DNA sequence, as you move along the chromosome, the chromosome isn't all the same, the DNA isn't all the same. It's actually packaged into entities or discrete units called genes. And there are other types of DNA present as well that perhaps give instructions like transcription factors and so on, and starting sites and stop codons. So there are little bits of DNA that are used for instructions. But they're normally, I suppose, made up as part of a gene. You also have bits of DNA that don't code for any genes at all, that are junk DNA. And actually it's in those parts of the DNA that can be very useful for studying evolution as well, and from which the microsatellites, for example, that I'll talk about later, are designed. But let's say if we're talking about say the functioning and the development of the organism, well then genes are very important, because genes are pieces of DNA that code for proteins. So each gene codes for a different protein, and then these proteins can either act alone or they can act together to help basically perform cellular functions, but also make up the bulk of the organism. So you have muscle tissue, hair, and skin, and so on, which are all kind of made up from proteins. So DNA is responsible for making proteins, and then proteins can actually go along to make up the phenotype of the organism. So the phenotype is the physical appearance of an individual. So what we see. And so they might be also defined as these observed and measurable traits that we can, so the wing structure, the color, the shape, the amount of muscle mass, the size of a muscle, and so on. So these would all be the phenotype. So we've got the genetics, so the DNA codes for the proteins, and the proteins help, I suppose, the organism to function, so the behavior, the metabolism of the organism, and of course the structure. And in between the genotype issuing the directions and the proteins building the structure and operating the organism, you have the environment. And so the effectiveness or the fitness of an individual, its genes and the proteins it makes, can actually be variable depending on the environment. The environment can have a very big role to play. And this is where you bring in natural selection. So Darwin's theory of natural selection is actually comes to the crux of this relationship between the genotype and the phenotype. So if we think about evolution as changes in inherited traits over time, we can actually use this, then we can use compare these traits over time to look at relationships between organisms. So this schematic here on the right kind of shows that in some detail. This is a very simple, what I call a phylogenetic tree relating 4 populations, A, B, C, and D. And if you look at the very top where the common ancestor is, here at the very top, what we see is that you have basically over time, you can have divergence happening within a species. And say you have 2 populations that don't interact for a period of time, so they're not reproducing, they're not producing offspring shared between the populations. So they're what we call reproductively isolated. And what happens over time in these isolated populations is that DNA divergence will start to occur. So you'll have mutations, mistakes happening in one population that don't kill the organisms, that actually seem to confer an advantage perhaps. And so therefore the DNA errors happens in those populations might get maintained. And so over time you get divergence between the 2 populations. And then over time, because DNA codes for protein, over time, if those populations remain separated, you get an accumulation of protein differences. And then that translates into functional differences, behavioral differences, and anatomical differences. So as long as these populations remain reproductively isolated, you start to see visually anatomical changes, you start to see behavioral changes. For example, you might get the emergence of mating calls or mating behaviors that then, if those populations do meet, that they no longer recognize the mating signals or the mating calls of each other. You might have different behaviors evolving to allow one population to exploit a new resource that the other population doesn't get to do. So you start to see these differences, and these are what we can actually also measure. So natural selection, the idea behind this is that in any population, in any species, you have multiple populations, and in any population you have variation. So individuals are different. They're not all exactly the same. They're not clones of each other, because mistakes happen in DNA. You also have other mechanisms in sexually reproducing organisms that increase genetic diversity. So populations vary, and then in any population, some individuals will be better able to survive, better at getting mates, and better then at leaving offspring in the next generation than others. So there's competition quite often. In most cases, there are more individuals produced than an environment can reasonably sustain, and therefore you get competition between individuals. And some individuals are more competitive than others and leave more offspring. So providing the variation that's contained within the population is genetically controlled, so that it's inherited by DNA, then the reproducing individuals will pass those traits on to the next generation. So if there are traits that are just accumulated within a lifetime of an organism, so for example, you've got parents that are very fit and very into exercise, and offspring are very muscular, the next generation won't necessarily be muscular unless they are genetically programmed to be more muscular than others. But generally just pumping iron and having lots of protein is not going to be inherited. And it's the same with equivalent traits in bees. So if you have variation in a population, some individuals are fitter than others and survive better and reproduce better. If those traits that help them to do that are inherited, then because they produce more offspring, those offspring will contain those traits, and therefore you start to see more and more of these traits in the population, and this is called adaptation. And this is reflected in the two pictures that are used here. So you have the dark bees in Northern Europe that are quite different from Ligustica bees in Italy. So variety is really important. So this genetic variation present in populations, and that's really one of the things we need to protect in bees, is their genetic variation, because it's really important for their health and fitness. So firstly, it increases this adaptive capacity. So the more variation you have in a population, the better that population is at adapting to change. And the kind of change that can happen might be geographical change. So you can get, so for example, climate change, global warming. You can have the impact of humans dramatically altering a habitat. Things like that, that if you have no variation in a population, then sometimes species can go extinct because none of the individuals in that population can adapt to new geographical changes. Also, you need variation in order to allow this competition to happen to increase fitness. So if you have in a population no variation, and some new individuals from a completely different population get brought in, they may outcompete the existing population very well if the existing population doesn't have the variation that allows them to compete successfully with these new incomers. You also have the situation with pathogens. So there's always a threat of new pathogens arising, and genetic variation is really important for that. In our case, part of our immune system, part of the immune system is highly variable. Some of the genes that are involved in our immune system are highly variable, that allow us to have a much broader response to new bacterial and viral pathogens. So some people in the human population might be more naturally resistant to COVID than others, for example. So some might be more susceptible, and it's shown that some DNA changes or DNA characteristics in some people might make them more susceptible to COVID. And it's the same with bees. So if you have a very non-variable population of bees, and you have a new pathogen, then it might be likely that all of the bees will succumb. Whereas if you have a very variable population, the likelihood is perhaps that some of those bees might have some traits that allow them to withstand that disease. For example, the introduction to Varroa certainly decimated populations of bees in Ireland, but it is feasible that because of the variation that was there, and because we had bees living in the wild at the time, that it's quite feasible that some some bees living in Ireland contained some traits that allowed them to be less susceptible to Varroa, and over time that these have developed resistance. And this is really important and is really of interest, obviously, to everybody to find bees that are resistant to Varroa. But certainly if we decline, if the numbers of bees decline to such an extent, or that we overuse particular strains of honeybees and we really reduce genetic variation, this will dramatically impact the honeybees' potential for coping with new challenges. And as well as, I guess, the variation being important for natural selection, genetic variation is also then important for us to be able to artificially select for traits that we actually like. So, this is very common in cattle and sheep, pigs, dogs, particularly, and horses and so on, but we also do it for bees. And some bees have been very highly selected in some countries like Germany, for example, where they really carry out extensive breeding on Carnica. But there are traits that beekeepers like honey production, docility, the color, and so basically beekeepers will artificially select for those traits. Okay, so if we come to the idea that understanding about basically we have evolution happening and then we have genotype and phenotype, and the environment, I said, was important in between those two. And we have to think that environments are very different in different places. So, and the environmental features of a particular location in one place might be, if bees are well adapted to that environment, if those bees are moved to a very different environment, the traits that made them so highly adapted to environment A might actually be detrimental in environment B. So, but the example here is that in the top pictures you see outside my house in November, dark, dreek, cloudy, rainy, windy all day, miserable for bees. And this can be fairly common in my neck of the woods. And the bees in Northern Europe have adapted to the cold and damp and weather and windy weather by being dark, and they apparently have other morphological traits that help them, for example, wind the muscles to help them fly in kind of windier weather. Whereas in the pictures in the south, you have the sunny climate in Italy, and you have brighter bees for reflecting the heat rather than absorbing heat. But traits that evolved in Apis mellifera mellifera, so the bees in the top, that help it thrive in these cold, wet climates, could be detrimental in warmer climates. And similarly, the traits that have developed in Apis mellifera ligustica that help it thrive in Italian climates might be detrimental in Ireland, so that they might not survive winter, for example, and might not survive very well, and might not do very well in summers, for example, that might be cold and wet. So over time, evolution has taken place on the ancestral, even the ancestral honeybee. And the honeybee, Apis mellifera, the Western honeybee, has actually evolved into quite a number of very distinct forms over this very large distributional range. So this map shows you the distribution range of the Western honeybee, Apis mellifera, and the different colors in the different places in the map reflect races or subspecies that have evolved. And this comes about because we have geographical separation of some places. So mountain ranges, for example, north of Italy into Switzerland, for example, you have the Alps, and this was a natural barrier for honeybees. And so bees in the south have evolved into Ligustica. And then north of that, we have the range of Apis mellifera mellifera. In orange here, we have the range of Carnica bees, and then we have other ones like Apis mellifera iberiensis and so on. And so in different parts of Europe where bees become isolated to some degree from other parts of Europe, and because honeybees have a very tight association with the environment, in terms of foraging and nesting and all of this, well, they then have evolved to adapt to these local conditions, and over time they have changed, and we can see these changes. So they're kind of defined as subspecies or races. And I have this slide in here because I just think it's kind of a natural conclusion to some of this evolution. So if populations become separated geographically and ultimately reproductively isolated, so the populations are no longer interbreeding, something called speciation can occur. And this is the production of a new species from an ancestral species. And so, for example, the picture on the left is the Grand Canyon. And this would have effectively separated, it started off maybe as a stream, and then erosion and so on has made it into, in some places, very, very large distance between the two populations that would have been on the top of that canyon on either side. And because over the years and years have been geographically and reproductively isolated, many, many species, the ones that can travel across the Grand Canyon, have evolved into separate species. So this is where you get the development of, as I said in the schematic, you have DNA divergence over time leading to protein divergence, and then leading to even behavioral and anatomical differences. And this cessation reduction of gene flow eventually can lead to the formation of completely distinct species that have shown here in the genus Apis. And if the barriers to reproductive isolation break down before the bees have evolved completely different reproductive systems or mating signals in order that they don't recognize the other population anymore as being the same species. So if the barriers break down before that happens, then speciation won't happen. So you continue to have interbreeding, and you might have the evolution of distinct races, and then they will go away again once hybridization mixes it all up into one single species. So we're interested in Apis mellifera on the right-hand side there. And within Apis mellifera, as I said already, there has been the evolution of these distinct races, or now what we call subspecies. So these are reproductively separated populations of the Western honeybee that have become adapted to the environment in which they're in, and they look and behave slightly different from each other. So this paper by Ayalisov into 2020, I can't pronounce it, but this is the latest paper I came across, suggests there are 33 different subspecies of Apis mellifera separated into 5 major lineages that have been isolated for a long time. And these subspecies, most of them we can distinguish via morphology and DNA. Number 14 on the top there is the northernmost distributional range of the Western honeybee, and that is Apis mellifera mellifera. So it's the natural range of Apis mellifera mellifera, and it includes Ireland. So what would happen here, I mean, it's quite interesting, because if these subspecies remain isolated, then they could eventually evolve to become completely reproductively isolated, i.e. separate species. But and what's happening now more recently by human interference, by bringing bees around the place, we are breaking down the natural barriers between these subspecies. And before this complete reproductive isolation is made in place, then the subspecies can intermix again and not be as distinct subspecies. So this is what's happening in some countries, whereby you have complete hybridization, different subspecies being brought into an area where it doesn't naturally belong, and so either replacing the existing subspecies or interbreeding with it. And this is, I guess, an issue for Ireland that we need to think about. So is there reproductive isolation between these subspecies? Or why am I going on about this? If we've got one species and we're moving it around and it's hybridizing and it's still one species, then what's the issue? And I think these two papers are quite interesting, because they suggest that there's some reproductive isolation behavior exhibited by Apis mellifera mellifera, which is the subspecies native to Ireland. So the top paper is from Poland, and they did a very nice study. In Poland, Apis mellifera mellifera and Apis mellifera carnica are both naturally present, and there is a hybrid zone between them, but they remain naturally distinct. So these authors got queens of each of the subspecies, and they artificially inseminated them with semen from each of the subspecies. So they took a mellifera queen and they artificially inseminated her with 50% sperm from mellifera and 50% sperm from carnica. They took a carnica queen and did the same thing. And then they looked at the offspring, and what they found was the Apis mellifera mellifera queens tended to only use mellifera drones, whereas the carnica queen used, the offspring were 50% by carnica drones and 50% by mellifera drones. And this suggested that there's reproductive isolation happening, and that the reproductive isolation seems to happen, is more strong in Apis mellifera mellifera than in Apis mellifera carnica. And then there's a second paper from Tasmania where they showed, this time it's between Apis mellifera ligustica and mellifera. And they showed that in the mountainous areas, cool, damp regions in Tasmania, then that there was reproductive isolation whereby Apis mellifera tended to use the drones of mellifera. And they suggested that the drones of Apis mellifera ligustica were less fit, and hybrids were less fit in the cool mountainous areas, whereas down in the hot other areas on the flat of Tasmania, I guess in environments that might be more similar to what Ligustica is used to, that Ligustica drones were fitter, and therefore you had more hybridization happening there than you had in the mountain regions. So it's kind of interesting that in Ireland, it could well be that we have Apis mellifera is native to Ireland, and if Apis mellifera is fitter in in comparison to the bees that tend to be imported, well then Apis mellifera might do okay despite imports. For example, if drones of mellifera are fitter in most cases than drones of imported bees. So in summary for this, I just wanted to say that basically mutations happen when DNA has been replicated, and it has done over the generations of honeybees in Western honeybees, since that ancestor of Western honeybees. And as the Western honeybee expanded into all of its distributional range in different environmental scenarios, we have the populations that are basically colonizing these new areas are exposed to new environments and challenges, and that this is a continuous process. So mutations are happening in the populations, creating variations. We've got variable populations of honeybees expanding their range into new environments. And this basically, as they expand into new environments, they become adapted. Those who can and who are fit in those environments become adapted to those local environments. And this, over time, as the bees extend out, has led to the formation of different races and subspecies and unique variants. And that basically the local variants that are present in these areas are probably the best adapted to those particular areas. And this is why we currently have a single species of honeybee, but more than 30 different subspecies across the distribution range, and why I suppose people are interested in local bees. Thank you.